Hi, I'm Eva Holtkamp. I'm a member of the Stegel group at the DKFZ in Heidelberg, Germany. And today I will talk to you about my master's thesis project, DeepRVAT, a method for joint modeling of rare variant genetic effects using deep learning and data-driven burden scores. I worked on this project together with people from my group, especially my supervisor, Brian Clark, who made great contributions to this project, and our collaborators from TU Munich. So, rare variants are expected to have strong phenotypic effects, and therefore studying them is of great relevance. However, the analysis of rare variants poses critical challenges. For example, we face data sparsity, and also a very high number, i.e. millions of different variants when analyzing large-scale cohorts, since many of the variants are private, so only present in a single individual. This leads to the problem of a high multiple testing burden when association testing would be done for each variant individually as in classical GWAS. And also, it is difficult to prioritize variants. So, existing RVAT methods address these challenges by performing region-based tests instead of individual variant tests, and by using functional variant annotation to inform variant prioritization. For example, in burden tests, potentially harmful variants are first identified using variant annotation-based filtering criteria, and then the number of harmful variants in a region, for example, gene, is counted to derive a gene-level burden score, and then to test for each gene if the burden score in cases is significantly larger compared to control samples. More sophisticated variance component or kernel-based tests perform region-based variance estimation, where the weight matrix allows to consider variant-specific effects as some fixed combination of functional variant annotation scores. Still, these conventional methods suffer from some critical limitations, which we aim to address with our DeepRVAT framework. First, standard methods only use annotations for filtering or within fixed, manually defined weight schemes. Here, we therefore utilize a multitude of existing annotation tools to capture diverse aspects of variant function. Second, since existing methods impose a strong filtering for harmful variants only using arbitrary filtering criteria, we want to leverage signal from high and moderate impact variants. Also, in contrast to existing methods, our approach is designed to account for nonlinear interactions. And finally, we do not only consider each gene in isolation, but train our model using signal from multiple genes. So let me introduce you to our framework. For training our notably trait-specific scoring models, we first of all select a set of so-called seed genes, so genes with known associations to a trait we are interested in. And we annotate all variants with different annotation tools, such that each variant is represented by a vector of currently 24 scores. In the second step, we apply a model that models gene impairment by aggregating the annotation vectors, such that we get a single score for each seed gene and each sample. And we combine these seed gene scores linearly to derive a phenotype value, and train the model by optimizing the loss between the actual and the obtained phenotype value. During association testing, we then annotate the variants of all protein coding genes and take the scoring model from the training step, which is now pre-trained and frozen, to obtain gene impairment scores for each gene and each sample. And we eventually test for association between the gene burden scores and the phenotype individually for each gene using standard methods. In the scoring model, we can in theory use any model that is differentiable. However, we have settled on the set network inspired by the DeepRVAT architecture that uses a network to embed the variants individually in a way that shares parameters across variants. And then we aggregate these variant embeddings to obtain a gene embedding, which is passed to a second model to derive a single score. Importantly, this architecture can handle arbitrary variant set sizes and is invariant to permutations of variants. And also, this gives quite decent results at relatively small architectures. So let me show you some of our results now. We first of all evaluated our method through simulation studies, where we used whole exome sequencing data from the UK Biobank to simulate phenotypes from simulated causal genes at varying fractions of causal variants. And we found that our method, shown in red, shows improved power of a conventional burden test, especially when the proportion of causal variance is low. And the false discovery rate was always controlled. In a second set of experiments, we applied DeepRVAT to find associated genes for 5 quantitative traits using whole exome sequencing data from the UK Biobank. We demonstrated that our method discovers more significant associations than a burden study on the UK Biobank dataset, which even used more samples. And we found no evidence of systematic inflation of p-values returned by DeepRVAT, and thus concluded that our method is calibrated. Also, we found through enrichment analysis that new DeepRVAT discoveries are enriched for non-affected genes of related phenotypes. By comparing the DeepRVAT discoveries to conventional burden tests on larger cohorts from the UK Biobank, we showed on the one hand that DeepRVAT is able to make more discoveries than any of the other studies at smaller sample sizes, and that as we increase sample size, DeepRVAT results have a higher and higher replication rate. Finally, we have some preliminary results indicating that trait specificity is important, since we consistently got worse results for mismatched models, meaning that we used a scoring model trained on a randomly chosen phenotype A when performing association testing for phenotype B. In summary, DeepRVAT is the first method to use deep neural networks to learn nonlinear effects from annotated variants, which greatly increases power to discover gene trait associations. Our method is well calibrated and discovers biologically plausible genes, and our results hint at trait-specific effects of variants. So thank you a lot for your interest, and if you're interested, feel free to get in touch with us.